Hello, and I'd like to thank the organisers of the meeting for inviting me to talk today. I'm Jane Armitage, and I've worked for many years in the Clinical Trial Service Unit at the University of Oxford. So this session is called Trial Labs of the Future, giving examples from the lens and other trials. But really what I'm talking about is efficient, low-cost trials, because that's what we hope that the trials of the future will be like. So why do we need them? Well, we need more trials, and in particular, we need better trials. And one of the ways that we can make better trials, and indeed make them low costs, so that more are possible, is to really focus on the things that are needed to keep the participants safe and to answer the research question. In other words, paring away all the other things that are less important. And one of the ways of doing this is to use data that is already being collected for other purposes. So you can think of the key to low-cost efficient trials as being those that can be embedded in routine practice, and that may or may not also involve using routinely collected data. And that can be either for feasibility, recruitment into trials, follow-up, or adjudication. But the key thing is also to keep things simple. So we're very lucky in the UK with having a wide range of potentially valuable data sets for use in clinical trials. And these are sort of listed here. So we've got hospital episode statistics, commonly known as HES. We've got information about death and cause of death. We've got disease-specific registries, such as cancer registries. We've got audit programs. We've got GP data, although at the moment this is much less easy to access than some of the other data. And we've got data from the nationally run screening services, such as the retinopathy screening, breast and bowel cancer. So I'm going to use examples from three trials, the LENS trial, the ASCEND trial, and the recovery trial. So LENS is embedded in the Scottish retinopathy screening service, and is, although I'm a co-I with David, it's really David Price who is both the brains and the drive behind this trial, which he initiated and got funded whilst he was still working in Scotland, and then, as it were, brought to Oxford when he came to work with us in our team. I'll also talk about the ASCEND trial, which I led with my colleague Louise Bowman, and the recovery trial, which, as many of you know, has been established by Martin Landray and Peter Horby, and I've not been involved, but it's been run within my group and by colleagues of mine. So I'll use this diagram to describe the various different trials. So we can use routinely collected information at various stages in the trial. And for LENS, it was used both for invitation and outcome data. I'll just give you a little bit of background about ASCEND. So it's a trial which is looking at the value of fenofibrate for diabetic retinopathy. And as many of you will know, retinopathy for people with diabetes is a major concern and a major cause of impaired vision and visual loss, and something that worries people with diabetes a great deal. In Scotland, but obviously also in other parts of the UK, we have a retinal screening programme, and typically that will involve taking a retinal image every 6 to 12 months. And if the retinopathy has felt to have progressed to a significant level, then such patients get referred to an ophthalmologist for further management. And often the retinopathy can be arrested by various treatments. So the question is, does treatment with fenofibrate slow or reduce the progression of diabetic retinopathy. And the reason this is of interest derives from the post-hoc analysis of two particular randomized trials. So the field trial, which was run in Australia and in Scandinavia, randomized about 10,000 people, or 9,500 people with diabetes. And across the whole trial, they saw that fewer of the participants who were randomized to Fenofibrate required laser treatment for diabetic retinopathy, as you can see here, 164 versus 238. And in a more detailed sub-study, they again saw reductions among those who were on Fenofibrate. A second trial, the Accord trial, which was actually a factorial design trial and randomized about 10,000 patients, had an eye sub-study. And again, they saw a reduction among those who got Fenofibrate compared with those on placebo, with a hazard ratio of 0.6 and a p-value of 0.006. But these were tertiary outcomes in both these trials. And importantly, the findings have not really led to changes in guidelines or uptake by clinicians. And we really need more data to see whether these were just chance findings, which seems unlikely given the p-values, but they are based on relatively small numbers. So let's just look at what happens when you have retinopathy screening. So you take a picture of the back of the eye, the retina, and from that you can grade the degree to which the diabetes has damaged the eye. And this is known as, so there's retinopathy grading. So the lowest grade other than 0 is background retinopathy, and then there's observable background retinopathy. So the background can either be mild or observable. And then you can also have problems with your macula, which is the central bit of where you actually see your central vision. And if that is damaged, that also has a major impact on your sight. So the primary endpoint for the lens trial is referable retinopathy. So if it progresses to such a state that you need to be referred to an ophthalmologist. So people are eligible if they've got any type of diabetes and some degree of retinopathy of the grades, either the lowest grade of maculopathy or an R2 grade or a bilateral, so in both eyes, the grade R1. So it's placebo controlled, double blind, and as I indicated, the primary outcome is progression to referable disease. And it is run, as I indicated, through the Scottish Health Board screening service. So there's a single screening visit conducted by a nurse who and the patients go into an active run-in with Fenofibrate. Fenofibrate is not entirely safe. It's a very generally well tolerated. It's been around for many years, but it does have an impact to raise creatinine levels, which is probably a reversible effect, but has an impact on the estimated GFR. It can also cause liver problems. And so there are considerations when running a remote trial, but the drug is mailed to the participants and all the follow-up, both using the routinely collected information, but otherwise is by telephone, mail or email. The sample size suggested that about 1,060 patients were required over about 4 years to see what might be plausible effects. And the estimates were there were somewhere between 20 and 30,000 people who might be eligible across Scotland. So that meant that about 4 to 5% of those people needed to be recruited into the trial. So there were two main routes. One was to identify the people who were already eligible through the Scottish Diabetes Research Network and invite them to take part. And then for people coming through the retinal screening service, if they were, they're sent a letter a few weeks after their screening, and if they were eligible, that letter could include an invitation to take part in the trial. So the patients, once randomized, and I'm pleased to say that they have now randomized 1,100 patients, actually just 1,150 patients. And those patients are now in follow-up. So I don't have any results to show you, but the linkage for the outcomes, as I indicated, is through the retinopathy service, through ISD, so the Information Services Division, which will give information on deaths and hospitalizations. And importantly, Sky Diabetes allows access for these particular patients through the appropriate approvals to access their biochemical data and safety blood results. So by linking to information that's already collected, it means that this trial can be run very cost effectively and very efficiently. So it means that the progression rate is well measured, and the patients have been involved in the design and planning of the study, and it's got the support of the RNIB. So it's a highly cost-effective trial being run to very high standards, and the hope that if the Fenofibrate clearly is reducing the retinopathy, then this will the data will be submitted for consideration of a change in the label. So I'll move now to the ASCEND trial, and for the ASCEND trial, which as I say, I led with my colleague Louise Bowman, we used routinely collected data both for the invitation into the trial, the outcome data, and we're currently using it for the post-trial follow-up. So the question that we were addressing in ASCEND is again in people with diabetes, but the question was, should people with diabetes who had not yet any clinical evidence of cardiovascular disease, should they be prescribed aspirin routinely? So we know that aspirin, once you've had a cardiovascular event, is associated with net benefit. But of course, aspirin does increase the risk of bleeding. And the question is, in these more moderate risk patients, does the benefit of aspirin outweigh the cardiovascular? Do the cardiovascular benefits outweigh any bleeding hazards? But of course, the challenge for us running this study was money, as always. We were very grateful and lucky to get British Heart Foundation funding to run the trial, and Bayer, who make the aspirin, agreed to provide aspirin and placebo free of charge, and also contributed towards the costs of the drug packaging. It was a factorial design trial, but I'm not going to talk about that. But with fish oils, and the fish oil manufacturers also provided the drug and some funding towards the cost of drug packaging. And the challenge really is that based on prior information, we needed to be able to detect what might be a plausible risk reduction, which was quite modest, of the order of 12 to 15% based on the other trials. And the sample size estimates originally suggested that 8,000 to 10,000 patients randomized and followed for 5 years might be sufficient, but it obviously depended on the event rate within the trial. But once we started recruitment, actually the event rate was substantially lower than we had initially estimated in our sample size estimates. And so we ended up planning for a 15,000 participant trial and follow-up for 7.5 years. And so the challenge was to try, how were we going to recruit 15,000 patients with diabetes from across the UK? And we actually, again, used the diabetes screening registers that were held generally locally for invitation to people with diabetes to attend their screening appointments. It needed quite complex levels of approvals through what was then called the Confidentiality Advisory Group. But with all the relevant approvals in place, we were able to get the names and addresses of people with diabetes sent to Oxford, the coordinating centre, so that we could send out invitations to people to take part. The Confidentiality Advisory Group also asked us to explore other means of finding patients, and so we also accessed GP diabetes registers, but in that situation we didn't have in the coordinating centre the names of and addresses of the patients up front, but we provided to the practices sort of invitation packages so that they could send those out to people with diabetes on their lists, and then there was a free post envelope so that they could return their questionnaires to our coordinating centre. And as you can see from this table, in total, if we look at the central registers column, we ended up inviting about 300,000 people who'd been identified through these centrally held registers, and randomized about 9,000 patients from those invitations. About 120,000 people were invited by their GPs through us providing these invitation packages, and about 5% of them ended up being randomized. And so that accounted for the bulk of our 15,500 patients who ended up being randomized. And essentially the trial was run entirely remotely. So again, very cost effective. We had simple eligibility criteria. We had forms, questionnaires that we sent to the participants for them to complete. And we backed that up with a 24-hour free phone service, which was manned generally by the administrative staff, but there was access to clinical staff if need be. And obviously the drugs and medication were also mailed. And you can see here the little white tablets are the aspirin or placebo, and the big orange capsules, the fish oil or placebo. So we were also using sort of remote methods able to get blood samples from about 3/4 of the randomized patients by sending a kit in the post for them to take to their GP and get a sample measured. And that allowed us to characterize the patients rather more in rather more detail than was possible just from their questionnaires. So we used routine information to supplement the outcome information. So we got outcome information. So we were obviously counting cardiovascular events, strokes, MIs, TIAs, and so on, and deaths. But we also were very interested in bleeding because of the risks from the aspirin. So we sent questionnaires to the patients. If we didn't hear from the patients, the GPs got questionnaires. We linked to the cancer registries, and for those patients who we were not able to get a follow-up form from either themselves or from their GPs, we then used the hospital episode statistics in order to ensure that we got sort of maximal follow-up. And of course, we got death notification from what was then ONS. So we ended up randomizing 15,500 patients, and I've sort of explained this, but by using these methods, we had over 99% complete information for both morbidity and mortality, with a mean follow-up of 7.5 years. I'll just show you the results just for completeness. We were able to demonstrate a 12% reduction in the risk of serious vascular events. So that was MI, stroke or TIA, or vascular death. And that is shown on a forest plot there. And if we also included revascularization, it was also 12%, reasonably highly significant reduction. But of course, I'm not showing you this now, but we did also see about a 40% increased risk of bleeding. And when we look at the balance of the benefits and the harms, it's not clear really that these low-risk patients, actually the benefits of aspirin really outweigh the risks. So one of the questions, of course, is to what extent can we rely on HES events, so from the routinely collected data, compared to sort of properly adjudicated events in clinical trials. And so in Ascend, we were able to do this because most of our events were actually reported by the participants or by their GPs, and we confirmed the event through the GP, so that they were clinically, the vast majority of them were clinically adjudicated events. So we could look and see what would have happened if we had only had access to the hospital episode statistics. And these are the results just for the majority of the patients. It actually excludes about 1,000 patients who were randomized from Scotland, although we have subsequently been able to get linkage for those Scottish patients. But this data is actually based on about 14,500 patients. And as you can see, this is what the adjudicated data told us, so that we saw this in this subset only an 11%, a hazard ratio of 0.89. But if we had just had access to HES or for these same patients, we would have got almost exactly the same result. Not quite, the confidence intervals are a little wider. And we've estimated that you might, if you only had access to the HES records, then you might well need to supplement these by having a slightly larger sample size. But it's very clear that actually, in general, HES gives you pretty much the same answer as using clinical adjudication, and that's a very encouraging finding. So I'm just going to finish in the last sort of 5 minutes with the recovery trial. I'm going to skip about Orion 4, which I had hoped to have time to do, but I will not do that. So I expect most of you have heard of recovery. So when the COVID-19 pandemic from SARS-CoV-2 emerged at the beginning of 2020, it was recognized very early on that this was a deadly infection, and reliable evidence was needed about what treatments might work and improve the clinical outcomes. And the trials needed to be unbiased, large enough to really get a robust answer, and that meant getting complete follow-up. And importantly, given that this was being run during the pandemic, it was very important to avoid an extreme burden on the local site staff. So my colleagues, well, particularly Martin Landry, but working with Peter Horby, but Martin's experience of running large, streamlined, randomized trials came to the fore, and he and Peter together designed the trial, in particular using routine data for a collection of outcome data, and that's what I'll show you, but it will allow post-trial follow-up as well. And obviously invitation is not relevant for an acute trial. And so they set up a sort of platform trial, if you like, and this trial in particular addressed hospitalized patients with COVID, either confirmed diagnosis or suspected diagnosis. And to begin with, several treatments were assessed, and if a patient was eligible for one or other of those treatments, they may get one or more of those treatments. It was an open label design, and so once randomized, patients could be prescribed the drug that they'd been allocated. And the primary outcome was death from COVID within 28 days of randomization. And so because this is such an unequivocal endpoint, there were no real anxieties about it being open label. And essentially what that meant was an adaptive design, and if a treatment was shown to be clearly beneficial, then that arm could be stopped. If it was not showing any benefit or showing harm, then that arm could be stopped, and other treatments have been added since. So it's an adaptive design with regular review by the Data Monitoring Committee. And at one point, the Data Monitoring Committee were meeting every couple of weeks. So tremendous contribution from them, led by Peter Sandercoe from Edinburgh. So the trial was designed to be have a simple informed consent and minimal data entry, online randomization, which took less than 5 minutes for the ward staff, a single case report form, either at 28 days or at discharge or at death. And then all the other information about patients was obtained using linkage to the routine data. So there was routine data, as you can see here on the left, the research data, the case report form, and these were collated by the team in Oxford, in particular led by Richard Haynes, Marion Maffam, and various colleagues. So what sort of data sets did they use? Well, you can see here quite how many were available, and indeed how many were actually used and are continuing to be used in the recovery trial. So there were hospitalization sets, data sets from England, Wales and Scotland, mortality data sets, as you can see there, disease-specific ones, particularly for cancer and renal registries, primary care data sets, which were allowed to be accessed because of the pandemic, but that has not so far gone, it's not at the moment been propagated beyond the pandemic. Then there were critical care data sets and obviously COVID specific data sets. And I'm not going to provide you with any results from recovery, but really just to show how this is possible. And of course now, so far they have randomized over 42,000 patients to a variety of different outcomes. Many of the arms have stopped. We know that both the steroids and the tocilizumab have shown benefit and on all-cause mortality and on some of the other outcomes as well. So a tremendous achievement that has had a global impact by allowing the trial to be embedded in very much in routine practice, minimizing the burden for the site staff and using the data that was being collected routinely, including gathering the baseline characteristics. So for example, the degree to which the diagnosis was confirmed, the ethnicity of the patients, and so on. So that, again, minimizing the amount of work for site staff, and there will be long-term follow-up going out to 10 years so that we can understand some of the longer-term effects of these treatments. I'm not going to go through the data management, but it is complex. I mean, for example, and I just picked out one statistic, there are 9 data sources that give a date of death. And so you obviously have to set up algorithms to ensure that these are checked and that there's a hierarchy and that you make sure that the most appropriate date is chosen. So it is a complex process and has required a team of people. It's been a challenge. There have been lessons that have learned through running of recovery. And these are just some of them, but the consent needs to cover the data linkage activities, the application processes for accessing data linkage are time consuming. I think we all know that, those of you who've tried to do this. There are implications for onward sharing of the trial data. And you really need to understand how the data is collected in order to ensure that the data management is appropriate. And informatics experience is vital to the success of this. The future challenges are around governance, the availability to researchers of the data, the validation of the outcomes, and the additional data sets and timely data flow. But I think that these are surmountable challenges to allow us to actually run trials efficiently and cost effectively using routine data. So thank you very much for your attention, and I hope that I have a chance to have a lively discussion with you at the end of the session. Thank you very much.